Welcome to this session on biochemistry. Today we are discussing on enzymes. So what are enzymes? Actually, enzymes are the magic molecules of life. These are the molecules. Actually, the enzymes are proteins, a kind of proteins, which are catalyzing the biochemical reactions. Biochemical reactions are considered as the most important part of maintaining life in this universe because all the characters are developed because of the production of different kinds of metabolites, and these metabolites are synthesized through biochemical reactions, and each and every step of these biochemical reactions are catalyzed by enzymes. So enzymes are considered to be the biological catalyst. They are the catalyst. Catalysts are those substances, those chemical substances which are actually controlling or triggering the chemical reactions, the speed of the chemical reaction. So if a chemical reaction is there, that chemical reaction should have an energy of activation. Suppose A and B are combining together, forming C and D. So A and B are considered to be the substrates, and C and D are considered to be the products. So A and B has to combine with each other to form B and D. So for combining A and B together, so there should be some energy of activation. Says some energy should be there associated with A and B to combine together, forming C and D. So that's the basic concept of chemical reaction. So that the energy which is required by the substrates to combine together to form the product is called the energy of activation, or simply it is called activation energy. So that amount of energy, that a minimum amount of energy is necessary with the substrates to combine with each other to form the products. That is simply called energy of activation or activation energy. So in most of the chemical reactions, the energy of activation is quite high, and in a normal condition, so these substrates cannot join with each other because the energy of activation is high, and the substrates will not have that kind of that much amount of energy available with them, so that the reaction will not happen. So catalysts are actually decreasing the energy of activation. So they are catalysts are chemical substances. Actually, they are not taking part in the chemical reaction, but they are actually lowering the energy of activation so that the substrates can easily bind with each other. The substrates can react with each other. So if two substrates has to react with each other, these substrates should have some energy of activation available with them. So if that energy of activation is quite higher, the enzymes can be utilized. So the enzymes actually will lower the energy of activation so that the enzymes so that the substrates can easily bind with each other to form the products. That is the duty of the catalyst. They are actually not engaging in the chemical reaction, but are involving the process of reducing the energy of activation so that the substrates can easily react with each other or bind with each other. So they are called a catalyst in a chemical reaction. So in a living system, there are chemical reactions. So suppose, for example, photosynthesis is a kind of chemical reaction. Respiration is a kind of chemical reaction. Fat metabolism is a chemical reaction. Okay, so all the life processes are associated with different kinds of chemical reactions, and these chemical reactions which are happening in the living system are totally called as biochemical reactions. Since those chemical reactions are taking place associated with the living system, they are simply called biochemical reactions. And in these biochemical reactions, again, there are catalysts. In the living system also there are catalysts, and those catalysts which are actually involved in the biochemical reactions are called enzymes. So enzymes are biological catalysts. They are catalysts acting in the biological system or the living system. They are catalysts which are actually engaged in the process of biochemical reactions. So they are simply called as biocatalyst. Enzymes are biocatalyst. Okay, so a large number of enzymes are there which are catalyzing each and every step of biochemical reactions. For example, in the case of photosynthesis, in the case of dark reaction of photosynthesis, the first reaction, ribulose biphosphate combines with carbon dioxide. So ribulose biphosphate and carbon dioxide are two substrates, and they are combining with each other with the help of an enzyme, and the name of that enzyme is Rubisco. So Rubisco is the enzyme which catalyzes the reaction between REBP and carbon dioxide, resulting in the production of 3-phosphoglyceric acid. That is the first stable product. Okay, so that kind of enzymes are there in all and every biochemical reactions in the living system. So we will call enzymes as biological catalyst. Biological catalyst. They are the magic molecules which are responsible for carrying out all and every biochemical reactions in the living system, and so they are generally considered to be the biological catalyst. Okay, the enzymes are proteins also. Enzymes are proteins. Enzymes are proteins. And all enzymes are proteins, but not all proteins are enzymes. Enzymes are simply proteins. They are catalyzing the biochemical reaction. So enzymes are proteins, and enzymes are the biological catalysts which are catalyzing, which are controlling the biochemical reactions in the living system. So they are supposed to be the magic molecules of life. Okay, then coming to the history, the study of enzymes is called enzymology. The study of enzymes is called enzymology. Okay, and coming to the history of enzymology, so it was this man, Kuhne, this man who coined the term enzymes. Kuhne, who coined the term enzyme, and the enzyme is actually a term which is derived from two Greek words, en and zyme. En and zyme. En means in, and zyme means yeast. In the sense, the first enzyme was actually derived from the yeast. The first enzyme was actually derived from the yeast. So the enzyme was the name enzyme was given to that particular kind of molecule, in the sense that the first enzyme was derived from yeast. En means in and zyme means yeast. So it is from these two Greek words the word enzyme is derived. Why it is called in yeast? Because the first enzyme was identified and isolated from the microorganism, the fungus yeast. So the name enzyme. Okay, and the first enzyme identified. Buchner was the first biochemist who identified the first enzyme from yeast, and the name of that enzyme is zymase. The first enzyme identified was zymase, and it was done by another biochemist, Buchner. So it was Kuhne who coined the term enzyme, and it was Buchner who first of all successful in identifying an enzyme from yeast, and he called the name that particular enzyme as zymase. Zymase. Okay. Then another biochemist, Subner, he was successful in isolating an enzyme, and the name of the enzyme is urease. So the term enzyme was proposed by Kuhne, and the term enzyme actually is derived from two Greek words, en and zyme. En means in and zyme means yeast. Then Buchner was another biochemist who was successful in identifying the first enzyme from yeast, and the name of that enzyme was zymase. Then another biochemist, Sumner, who was successful in isolating an enzyme from an organism for the first time, and the name of that particular enzyme is urease. This is a brief account on the history of enzymology. Okay. So coming to the structure of enzymes. So you know enzymes are biological catalysts which are catalyzing the biochemical reactions, within the living system. Okay, and you know enzymes are proteins. The major core of enzymes are proteins. Okay, coming to the structure of an enzyme. Okay. So when we are just studying the structure of an enzyme, the enzyme will have the enzyme will have two important components. The first component is called apoenzyme, and the second component is called prosthetic group. Apoenzyme and prosthetic group. Okay, what is apoenzyme? You know, so this is the enzyme. Okay, this is the enzyme. This is actually the enzyme. And this is the major part of the enzyme. This is the proteinaceous part of the enzyme. You know, enzymes are proteins, and this is the proteinaceous part of the enzyme. And this major proteinaceous part of the enzyme is called apoenzyme. The core structure of the core part of the enzyme is called apoenzyme. That is actually the protein. So the proteinaceous component, the major proteinaceous component of the enzyme is called apoenzyme. The major proteinaceous component of the enzyme is called apoenzyme. Okay, but in normal cases, in normal procedure, with only a proteinaceous component, the enzyme cannot work. So enzyme may need a non-protein part associated with it for its for its actual performance, for its actual function. So a protein with an enzyme with apoenzyme, only apoenzyme may not work because it is only the protein component, and that protein component may need the help of a non-protein part in order to function properly. And that non-protein part may not always may not always in tight bonding with the protein part. They may be different. So you can see. So this is the protein part. So this is the protein part. This is the apoenzyme. This is the major part of the enzyme, and this is actually the non-protein part. In normal condition, in an inert condition, so these two may be at different positions. These two part, this protein part as well as the non-protein part, will not be in tight binding. They will get separated. But in order to function properly, this protein part may need the association of this non-protein part, and this non-protein part of the enzyme is called prosthetic group. So an enzyme, a functional enzyme, may have two components: the major protein core part of the enzyme and a non-protein part. The major protein core part of the enzyme is called apoenzyme, and when that apoenzyme has to work properly, it may need the support of a non-protein component, a non-protein part, and that non-protein part or non-protein component of the enzyme is called prosthetic group. That non-protein part is called prosthetic group. Okay, so whenever we are just studying the structure of an enzyme, it will have a major protein component. A functional enzyme will have a major protein component and a non-protein component. The protein component, the major core protein component of the enzyme is called apoenzyme, and the non-protein part of the enzyme is called prosthetic group. Okay, you can see. So this is the protein component, and this is the non-protein component. For that enzyme to become functional, this non-protein component has to bind with the protein component. This is the functional enzyme. You can see this is the functional enzyme where the protein as well as non-protein components are joined together. So this is the functional enzyme. So this is the protein component, which is called apoenzyme, and this is the non-protein component, which is called prosthetic group, Okay, and this non-protein part may be of two different kinds: coenzyme and cofactor. What is the difference between this coenzyme and cofactor? Okay, coenzyme means it is the non-protein part. It is actually, coenzyme is the non-protein. So first of all, we have to study what is cofactor. Cofactor is actually the non-protein part, which is actually a kind of metal ion, maybe a kind of metal ion, a small group of chemical substances, for example FAD or aluminum. So some enzymes may require some metal components. So that kind of factors are called, that kind of non-protein part is called cofactor. Okay, and when coming to the coenzyme, that may not be a single metal ion, or it may be a non-protein molecule. That may be a non-protein molecule associated with the apoenzyme. So that is the difference. Coenzyme may be a large molecule, a large non-protein molecule that is get associated with the apoenzyme, and cofactor, it may be a simple kind of metal ion or something like that, which is just in tight union with the apoenzyme during the process of enzyme action. So that is the difference between coenzyme and cofactor. So coenzyme is a kind of prosthetic group, and cofactor is another kind of prosthetic group. Coenzyme means it is a large non-protein molecule. It's a large non-protein molecule which is get associated with the apoenzyme for and are acting as the prosthetic group. And cofactor is a kind of prosthetic group, which may be a small kind of metal ion or something like that. That is the difference between coenzyme and cofactor. So this is regarding the structure of the enzyme. Okay, so there is a protein part of the enzyme, and that protein part means it is the apoenzyme. It is the core part of the enzyme, and in order to supplement the activity of the apoenzyme, normally an enzyme may require a non-protein component, and the non-protein component is called a prosthetic group. It may be of two different kinds, coenzyme and cofactor. Okay, then when And this apo enzyme and prosthetic group are located at different positions when a reaction is at the inert state, when that reaction is not in an active state. When a reaction has to become active, so the enzyme has to become active. So for that, the apo enzyme has to bind with the prosthetic group. The apo enzyme has to bind with the prosthetic group. So at that time, when a reaction has to become active, the enzyme has to become active. And for that, the prosthetic group has to bind with the apo enzyme. When a prosthetic group gets binded properly with the apo enzyme, so that is the functional enzyme. Apo enzyme is the major protein component of the enzyme, core protein component of the enzyme. This is actually the enzyme. Apo enzyme is actually the enzyme, but it is not active. Okay, so to activate the apo enzyme, it may require the non-protein part, that is the prosthetic group. And when a prosthetic group is get binded with the apo enzyme, so that is considered to be the functional enzyme, and that functional enzyme is called hollow enzyme. That functional enzyme is called hollow enzyme. That functional enzyme is called hollow enzyme. Okay, so hollow enzyme is formed when associated cofactor or coenzyme binds to the enzyme's active site. So enzyme is a molecule where we can see the protein. The major part of the enzyme is the protein component, which is called apo enzyme, and it is the major part. But that apo enzyme will become functional only when a non-protein parties get joined with it. When a non-protein parties get joined with the apo enzyme, then that enzyme will become functional, and that functional enzyme is called hollow enzyme. So when we are discussing the structure of the enzyme, we should know what is an apo enzyme. This is the major protein component or not major protein part of the enzyme. Okay, then what is a prosthetic group? It is the non-protein part of the enzyme, which is very much essential for the proper functioning of the enzyme. It may be of two different kinds, coenzymes and cofactor. Okay, then when this apo enzyme and prosthetic group are just binded with each other, then it will become a functional enzyme, and that functional enzyme is called hollow enzyme. That functional enzyme is called hollow enzyme. So what is hollow enzyme, what is apo enzyme, what is prosthetic group, and what is the difference between coenzyme and cofactor? Okay, this is regarding the enzyme structure. Okay, then some variations, some differences, some terminologies of some different kinds of concepts in enzymology. Okay, the first one is endoenzymes and exoenzymes. This is what is the difference between endoenzymes and exoenzymes. So endoenzymes, in the sense, these are the enzymes that can be seen actually within a living cell. Okay, within a living cell. The enzymes which are acting inside the living cell is called endoenzyme. But there are some other enzymes that can be seen outside the living system, outside the living cell, not in the living system, outside the living cells. That means in between the intercellular spaces or something like that, we can see some kind of enzymes, and those enzymes are functioning, and those kinds of enzymes are called exoenzymes. So that is the difference between endoenzymes and exoenzymes. Endoenzymes are those enzymes which can be seen working within a cell, inside a cell, and exoenzymes are those enzymes that are working outside the cell, actually working outside the cell. They are called exoenzymes. Okay, then what are isoenzymes? Another concept is called isoenzymes. Isoenzymes are those enzymes which are having isoenzymes, in the sense, iso means similar. Isoenzymes means similar enzymes, in the sense similar enzymes. Okay, so isoenzymes are those enzymes which are actually catalyzing different kinds of enzymes. Isoenzymes means, suppose there are 5 different enzymes and they are catalyzing, they're actually catalyzing a single biochemical reaction. So these enzymes are called isoenzymes. Okay, so they are having isoenzymes are those enzymes which are having different structure. So it can be defined as an enzyme group with different structure but are catalyzing a single biochemical reaction. A group of enzymes which are having different structure but are catalyzing a single biochemical reaction. So they are called isoenzymes. Isoenzymes are those enzymes which are considered, they are coming under a single group and are having different structure but are catalyzing a single biochemical reaction, so they are called isoenzymes. Then ribozymes. What are ribozymes? In some case, sometimes ribosomes may be acting as enzymes. Ribosomes may be acting as enzymes, and when the ribosomes are acting, ribosomes are protein complexes. In some cases, sometimes those ribosomes, some ribosomes can act as enzymes, and when the ribosomes are acting as enzymes, they are called ribozymes. So endoenzymes means those enzymes which are working within the cells, inside the cell. Exoenzymes means those enzymes that are working outside the cell. Isoenzymes are the group of enzymes which are having different structure but are catalyzing a single biochemical reaction. And ribozymes means the enzyme. Sometimes the ribosomes, the cell organelles, non-membrane bounded cell organelles such as ribosomes can function as enzymes, and those enzymes are called ribozymes. Okay. Then coming to the classification of enzymes. So, different kinds of biochemical reactions are operating within the living system, whether it is plants or animals or microorganisms. So, a large number of biochemical reactions are there in all these metabolic activities, and each and every step of those biochemical reactions will be catalyzed by a biological catalyst called enzymes. So, a large number of enzymes are there in the living system. So, we have to classify those enzymes. Okay. So there is a universal protocol to classify the enzymes, and those universal protocol was proposed by the Commission on Enzymes of the International Union of Biochemistry. Commission on Enzymes, a general protocol for classifying the enzymes in a scientific way was proposed by the Commission of Enzymes on the International Union of Biochemistry in the year 1961, and just we are following this pattern for classifying the enzymes. Okay. As per the protocols of this Commission on Enzymes of the International Union of Biochemistry, suppose an enzyme can be named as just like this 1.1.1.1. 1.1.1.1. So it can be designed as, it can be titled as, it can be named as digits of four digit components or four class components. Okay, so 1.1.1.1, that is the name of an enzyme. Suppose there is another enzyme, it may be 2.2.3.4. So there will be 4 digits in each and every in the name of each and every enzyme. So this is the pattern proposed by the Commission on Enzymes of the International Union of Biochemistry in 1961. Okay, so what it denotes, what this 1.1.1.1 denotes. So according to the recommendations of the Commission on Enzymes of the International Union of Biochemistry, the enzymes are classified into six classes. At first, the enzymes are classified into 6 classes. Okay, class 1, oxidoreductases. Class 1 is and a group of enzymes called oxidoreductases. Class 2, another group which is called transferases. The class 2 of enzymes are called transferases. Class 3, hydrolyses. Class 4, ligases. Class 5, isomerases, and class 6, lyases. So these are the 6 classes of enzymes as per the rules of Commission on Enzymes of the International Union of Biochemistry. So there are 6 classes of enzymes. The first class is called oxidoreductase. Second class is called transferase. Third class is called a group of enzymes called hydrolyses. The fourth one, ligases. Fifth one, isomerases, and the last class of enzyme is called ligases. So the fifth class is always isomerases. So if we say that particular enzyme belongs to the fourth class, that means that kind of enzyme may be that may be an enzyme, that may be enzyme which is coming under the class ligases. It may be a ligating enzyme. If we are saying an enzyme as coming under the class 3, we should identify that enzyme as a hydrolyzing enzyme. Okay, if we say an enzyme is coming under the class 5, that means that enzyme may be an isomerizing enzyme. If you are saying an enzyme is coming under the class 1, That means we will identify that that particular enzyme is catalyzing is an oxidoreductase enzyme. It is an oxidoreductase enzyme catalyzing an oxidoreduction oxidative. That means a redox reaction. It is actually catalyzing a redox reaction. If we are considering an enzyme which belongs to the class 2, then definitely we will say that is a transferase enzyme. So just like we are just classifying the total enzymes in the living system into 6 different classes. Class 1 oxidoreductases, class 2 transferases, class 3 hydrolysases, class 4 ligases, class 5 isomerases, and class 6 lyases. So there is a shortcut for identifying the name of these classes. We should identify it as OTHLIL. OTHLIL. O stands for oxidoreductases, T transferases, H hydrolysases, L ligases, I isomerases, and the last one lyases. So OTHLIL, that are the 6 different classes of enzymes: oxidoreductase, transferase, hydrolyse, ligases, isomerases, and lyases. So that is the first step. So we will classify the entire enzymes in this universe into 6 different classes. Class 1 oxidoreductases, class 2 transferases, class 3 hydrolysases, class 4 ligases, class 5 isomerases, and class 6 lyases. Okay, then each of these classes are classified again into subclasses. So oxidoreductase is again classified into subclasses. Transferase is again classified into subclasses. Hydrolysis is again classified into subclasses. Okay, so classes, 6 classes, and each of these classes are again classified into subclasses. Subclasses again can be classified into subclasses. Each of these subclasses again can be classified into subclasses, and finally each of these subclasses can again be classified into subclasses. So first of all, we will classify the enzymes into 6 major classes. Each of these classes again can be classified into subclasses, subclasses into subclasses, and finally the subclasses into subclasses. So for example, if we are naming an enzyme as 1:1.1.1.1, that means that enzyme belongs to the class 1, oxidoreductase. Okay, and under the class 1 it belongs to the subclass 1. And under the subclass 1 it belongs to the subclass 1. And under the class 1 it belongs to the subclass 1. So if it is 2.3.2.3, then it belongs to the class 2. Under the class 2, subclass 3. Under the subclass 3, subclasses, under the subclasses, class 2. Again under the subclass 2, subclass 3. That is the way where we are just naming each and every enzymes according to the universal protocol proposed by the Commission on Enzymes of the International Union of Biochemistry. So this is regarding the enzyme classification and nomenclature. Okay, so there are some set of rules proposed by the Commission on Enzymes of the International Union of Biochemistry in 1961, and according to that we are just classifying and naming the enzymes. And according to this set of rules, there are 6 classes of enzymes. What are those 6 classes? Oxidoreductases, transferases, hydrolysases, ligases, isomerases, and lyases. Then classes are classified into subclasses, subclasses are classified into subclasses, and finally subclasses are classified into subclasses. Okay. And this is the mechanism of enzyme action. Just I have already mentioned. So these are the reactants. For example, if we have to combine carbon dioxide and water together, so these are the reactants. And when these reactants has to combine with each other, so there should be some minimum amount of energy, and this amount of energy is called energy of this amount. This much amount of energy is very much essential for combining carbon dioxide and water together, and this is simply called the energy of activation or activation energy. Okay, so without enzyme, the activation energy for combining carbon dioxide and water is this much. But when we are just adding some enzymes, when we are providing the assistance of enzymes to this reaction, what the enzymes are doing, these enzymes will lower the energy of activation. This is the actual amount of energy which is required for carbon dioxide and water to combine together. But when we are providing the assistance of enzymes, these enzymes will lower this activation energy into this level, so that the reaction can happen in a very efficient way, in an easy way. If this amount of energy is required, this reactant has to get this energy from somewhere else, otherwise the reaction will not happen. But if we are providing the enzymes are actually lowering the energy of activation, so that the reactants can combine with each other, forming the products. Okay. Then coming to the mode of enzyme action, how the enzymes are catalyzing a biochemical reaction. So in a biochemical reaction, there may be the substrates. Okay, and the substrates are finally get converting into the products. Enzymes in a biochemical reaction, there are one or more substrates. For example, carbon dioxide and water. These are the two substrates on that particular chemical reaction. So they will have to combine with each other, and for effectively catalyzing that biochemical reaction, we are providing the assistance of enzymes. And how the enzymes are actually catalyzing the biochemical reaction. Suppose this is the mechanism. This is the route map which explain how the enzymes are catalyzing a biochemical reaction. Okay, so suppose this is the substrate. This is the substrate, and these substrates are just undergoing the chemical reaction and converting into products like this. So substrates are converting to products like this. How the enzymes are catalyzing this reaction? So this is the enzyme. Actually, this is the enzyme which is catalyzing this biochemical reaction. Okay, and there is a site. There's a specific site within the enzyme. There's a specific site within the enzyme to which the substrates are binding, and that area is called the active site of the enzyme. So the substrates are binding with the enzyme, not at every part of the enzyme, but the substrate will identify a very specific area within the enzyme to which it is get binded, and that very specific site, that very particular site within the enzyme molecule where the substrates are binding is called the active site of the enzyme. That area is called active site. So see, in this case, in this particular enzyme, this is the active site to which the substrates are binding. So substrates are coming, and enzyme is actually here. Enzymes will have an active site, and the substrates will come and bind with the active site of the enzyme. So this is a process. Substrate come and bind with the active site of the enzyme. So this here it forms a complex. So this is the enzyme, this is the substrate. During the chemical reaction, during the biochemical reaction, the substrates are coming and it is get binding with the active site of the enzyme. So that at that particular point, a complex will be forming, and that complex is called enzyme substrate complex. Enzyme substrate complex. So enzymes are there, substrates are coming, substrates are binding with the active site of the enzyme, forming a complex, and the complex is called enzyme substrate complex. Once the enzyme substrate complex is formed, what the enzymes will do, the enzyme will lower the energy of activation, so that the substrates are reacting with each other. And forming the products. Okay, so enzyme substrate complex then will be get converted to enzyme product complex. So this is the substrate and this is the enzyme. Enzymes, whenever the enzyme substrate complex is formed, the enzymes will lower the energy of activation, so that the catalysis, that process is called, whenever the enzyme is lowering the energy of activation, that process is called catalysis. When the energy of activation is get lowered, what will happen? The substrates will react with each other, forming the products, and these are the products. These are the products associated with the enzyme, and that is another complex. This is simply called enzyme product complex. And finally, the products will be get released, so that the enzymes will remain as such without any change, but the substrates are get changed into products. So enzymes and products will be released at the end of the biochemical reaction. This mode of enzyme action, this is the way by which the enzymes are catalyzing a biochemical reaction, and this mode of enzyme action was identified and proposed by another biochemist, Michaeli-Menten. This is simply called Michaeli-Menten's mode of enzyme action. Okay, and according to the model proposed by Michaeli-Menten, what will happen? The substrates are coming, and the substrates are binding with the active site of the enzyme, forming a complex, and that complex is called enzyme substrate complex, or it is otherwise called Michaeli-Menten complex. Enzyme substrate complex or Michaeli-Menten complex. Okay, whenever the enzyme substrate complex is formed, the enzymes will lower the energy of activation. That means the process of catalysis will happen, so that the products will bind with each other, forming the substrates will bind with each other, the substrates will react with each other, so that the products will be forming the enzyme product complex. And at the end of the chemical reaction, the enzymes will be released and the products also will be released. Enzymes will remain as such and the substrates are get converted into products. So this is a mode of enzyme action, and this was proposed by Micheli-Menten. Also this is called Micheli-Menten's mode of enzyme action. Okay. Then there are some theories. There are some theories which explain this mode of enzyme action. There are some theories which explaining the process of enzyme action or the mode of enzyme. So during the mode of enzyme action means enzymes are coming, substrates are coming, substrates are binding with the active site of the enzyme. Okay, the enzymes will lower the energy of activation or catalyzing the biochemical reaction, so that the substrates will react with each other, forming the enzyme product complex, and finally they will be get separated. The enzymes are released and the products also will be get released. So this is called the mode of enzyme action. This is simply the mode of enzyme action, or Micheli-Menten's concept of enzyme action. And there are some theories which support this Micheli-Menten's model of enzyme action. And the first theory is called lock and key theory. Lock and key theory. And this lock and key theory is proposed by a biochemist, and his name is Emil Fischer. This man, Emil Fischer. Lock and key theory proposed by Emil Fischer. Okay. So Emil Fischer compared the process of the mode of enzyme action, or the process of enzyme action, or the enzyme catalysis process with the lock and key mechanism. The lock and key mechanism. So so if you are having a lock, that lock can be opened by means of a key. So if you are having a lock, and that lock can be opened by only a single key. Every key cannot open that lock, but a single key can open that lock. If you are using a wrong key, the lock will not be get opened, and if you are using the right key, only the enzyme, the lock will be get opened. So the enzyme action is just like that. The enzyme action is just like that. So Emil Fischer compared the substrate with the key and lock with the enzyme. Substrate with the key and lock with the enzyme. Okay, that means this particular lock can be opened by this particular key. Only this key can open this lock. Just like that, only a specific substrate can bind with the active site of an enzyme. So you can see this is the enzyme and this is the active site, and this active site can be occupied by this particular the substrate with this particular shape. Okay, if you are having a substrate like this one, if our substrate is just like this one, it cannot bind with this active site. Only the substrate of this shape can bind with the active site of this enzyme. Okay, so the first step of enzyme action is the binding of substrate with the active site of the enzyme. And if that process has to become successful, some specific substrate only can bind with the active site of the enzyme. Just like a specific key can open a specific lock. Only the substrates with some specific structural conformation can bind with the active site of an enzyme, of the active site of the particular enzyme. For example, Rubisco is an enzyme. So the enzyme Rubisco will have an active site, and that active site is tuned to occupy carbon dioxide. Tuned to occupy carbon dioxide. If water is coming, the water cannot bind with the active site of the Rubisco, because the active site of the Rubisco is tuned to accommodate only carbon dioxide, not water. Just like that, every enzymes will have an active site to which a substrate with a specific structural conformation can bind. Okay, so this substrate only can bind with the active site of this particular enzyme, forming the enzyme substrate complex. Then the usual process will go on. The substrates will react with each other, forming the product, and the enzymes and products will be get released. The first step is so crucial, the formation of enzyme substrate complex. It can become possible. The enzyme substrate complex can become possible only if the active site of the enzyme can properly accommodate a substrate. Only some specific substrate can bind with the active site of an enzyme. So that key theory is called just like a specific key can open a lock. Specific key can only right key can open a lock. a wrong key cannot open a lock, or that means a suitable substrate can only bind with the active site of that particular enzyme. So this theory is called lock and key theory, proposed by Emil Fischer. So this is a theory to explain the mechanism of enzyme action proposed by Michaeli-Menten. And according to this theory, the mode of enzyme action is just like the operation of a lock and a key. The key can be compared with the substrate, and the lock can be compared with the enzyme. Only a right key can open a particular lock. Just like that, only some specific substrates with some specific substrate with a defined structural conformation can bind with the active site of the enzyme, forming the enzyme substrate complex. So this is the concept behind lock and key theory proposed by Emil Fischer. Okay. Then the second theory which explains the mechanism of enzyme action is called induced fit mechanism. Induced fit mechanism, and this theory is proposed by another biochemist, and his name is Koshland. Induced fit mechanism or induced fit theory proposed by induced fit mechanism or induced fit model or induced fit theory proposed by another biochemist Koshland. It is also to explain the Michaeli-Menten concept of enzyme action. Okay, and according to this theory, so these are the substrates and this is the enzyme. These are the substrate and this is the enzyme. Okay. Normally, the active site may not be suitable for accommodating this kind of substrate. So, according to the lock and key theory, this if the substrate with this kind of specific structural conformation is coming close to the enzyme, the structure is not suitable for this. The specific structure of the substrate is not suitable for binding with the active site of the enzyme. Now, according to the lock and key theory, this kind of enzyme action will not happen because the substrate is having a specific configuration and the enzyme active site is quite different, so that the substrate cannot bind with the enzyme. But according to the induced fit model, there are some cases where the substrate can modify the active site of the enzyme. So the substrates are coming and their structure is not suitable for binding with the active site of the enzyme, even though it is not suitable for binding with the enzyme. With the active site of the enzyme, the substrate can change the structure of the active site of the enzyme. So the substrates are coming and are inducing the enzyme to change the structure of the active site so that the substrate can easily bind with the enzyme. That's the difference. The substrates are coming and the enzyme is there. The active site of the enzyme is not suitable for binding with the substrate. So what the substrate is doing? The substrate is coming and it is inducing the enzyme to change its active site in order to accommodate the substrate. Okay, so that the enzyme is changing its active site. The enzyme is changing or forced to change the conformation of their active site so that the enzymes can bind properly with the active site. The substrate can bind properly with the active site of the enzyme. So enzyme substrate complex formed. Then normally the enzymes will lower the energy of activation so that the substrates will bind with each other, forming the product. The products are removed and the enzymes will remain as such with a normal structural configuration. So this is called induced fit model or induced fit mechanism proposed by Koshland. So induced fit model or induced fit mechanism or induced fit theory is another theory which explains the mechanism of enzyme action proposed by Michaeli-Menten. And according to this induced fit model, the substrate, the shape of the conformation of the substrate may not be suitable for the active site of the enzyme. But whenever the substrate is approaching the enzyme, the substrate is inducing the enzyme to change its active site in order to accommodate that particular substrate, so that the enzyme will modify its active site so that the substrate can bind with the active site of the enzyme. Once the enzyme substrate complex is formed, then normally the rest of the process will happen. The enzyme will lower the energy of activation. The substrates will be reacting with each other, forming the products, and finally the products will be released and the substrates, the enzyme will remain as such. And this is called induced fit mechanism. So there are two theories or two mechanisms to explain the Michaeli-Menten's concept of enzyme action. The first theory is proposed by Emil Fischer, which is called lock and key theory, and the second theory is proposed by Koshland, and the theory is called induced fit mechanism. Okay. Then what are the properties of enzymes? What are the different properties of enzymes? So you know enzymes are biological catalysts, and what are the properties of enzymes? The first property of enzyme is it is catalytic. Enzymes are catalysts, so they are having catalytic property. Catalytic property means the property which decreases the energy of activation so that the substrates can easily react with each other. So that property is simply called catalytic property. So enzymes are having catalytic property. Enzymes are biological catalysts, and so they are having catalytic property. Then enzymes have specificity. Enzymes have specificity. That means only some specific substrate can react with the enzymes. Only some specific substrate can bind with the enzyme. So enzymes are so specific. Enzymes are so specific. For example, Rubisco, the enzyme Rubisco can accommodate carbon dioxide. Enzyme phosphoenol pyruvic acid kinase can accommodate carbon dioxide. So just like that, enzymes will have specificity on the substrate. Some specific substrate can only react with the enzyme. So enzymes are highly specific. And most of the reactions, most of the enzyme catalyzed reactions are reversible. Forward and reverse reactions can happen. Most of the biochemical reactions catalyzed by enzymes are reversible. So it is because the enzymes have some kind of reversibility. So enzymes are having reversibility. Then enzymes are sensitive to heat. Enzymes are sensitive to temperature. That means the enzymes will work only at a specific temperature, and if the temperature is above that optimal level or below that optimal level, the enzyme will not work. Enzymes will work properly at a specific temperature, and that specific temperature where the enzyme action is maximum is called the optimum temperature. When the temperature is exceeding beyond that optimal level or decreasing beyond that optimal level, the rate of enzyme action will be get decreased, or sometimes literally there will not be any enzyme action if the temperature is above or below the optimum temperature. So enzymes are highly sensitive to temperature, and the enzymes are highly sensitive to pH also. The enzymes are highly sensitive to temperature and pH, and the enzymes are having a colloidal property. Enzymes are colloidal in nature. They are not simply liquids or solids in between them. The nature of the physical nature of the enzyme is in between them, and it is the colloidal. Enzymes are having colloidal property. So enzymes are catalytic, enzymes are having specificity, enzymes are having reversibility, enzymes are sensitive to heat, temperature and pH. The enzymes are colloidal, and finally the enzyme action is controlled by so many different kinds of inhibitors. Inhibitors in the sense there are some substances which are having the ability to suppress the enzyme action, to deactivate the enzyme action, to inhibit the enzyme action. There are some substances in the medium that can have the ability to inhibit the enzyme action, and those substances are called enzyme inhibitors. Those substances are called enzyme inhibitors. Okay. Then what are the different factors affecting temperature, enzyme action? What are the different factors affecting enzyme action? Temperature is a factor. Just already I have mentioned. There is an optimum temperature where the enzyme action is maximum, and below and above that particular temperature the enzyme action will be get suppressed. pH is another factor. pH is actually another factor which is affecting the enzyme action. The enzymes will be working on a specific pH. If an enzyme has to work on the pH 7.2, it will work only at 7.2, and if the pH is 7.5, that enzyme will not work. So this is another factor. The presence of water is another important factor which is affecting the mechanism of enzyme action. Then concentration of the substrate. As the substrate concentration increases, the rate of enzyme action increases, and that will reaches a maximum. For example, if you are having 5 molecules of enzymes and only 1 molecule of substrate, the enzyme action will be a minimum. If the substrate is 2, the enzyme action will be increased. The substrate is 3, again the enzyme action will be increased. Substrate is 4, increased. Substrate 5, that means substrate 5, enzyme 5, the enzyme action is maximum, and it will remain stationary. If the substrate is 6, there will not be further increase. So the concentration of substrate is another factor. If the concentration of substrate is increasing, the rate of enzyme action is increasing, reaches a maxima, and then remains stationary. Okay, then concentration of the enzyme is again another factor. Again, the concentration of the enzyme also will increase the rate of enzyme action. If the concentration of enzyme is increased, the rate of enzyme action again will be get increased, reaches a maximum, and then it remains stationary. Then enzyme inhibitors, the chemical substances which are some chemical substances in the medium can inhibit the process of enzyme action, and if that kind of chemical substances are there will not be any kind of enzyme action. Then accumulation of end products, if the final products of the biochemical reaction is get accumulated, that also will adversely affect the rate of enzyme action. So these are the different factors which are affecting the enzyme action: temperature, pH, water, concentration of the substrate, concentration of the enzyme, presence of enzyme inhibitors, then accumulation of the end products. Okay, then coming to the enzyme inhibitors, this is a major concern. Enzyme inhibitors means the chemical substances which are having the ability to inhibit or check the process of enzyme action. So there are some if there are some chemical substances in the medium, some chemical substances can inhibit or check the enzyme action, and those chemical substances are called enzyme inhibitors. And we can classify the enzyme inhibitors into three different groups: competitive inhibitors, non-competitive inhibitors, and feedback or allosteric inhibitors. Competitive inhibitors, non-competitive inhibitors, feedback or allosteric inhibitors. The inhibition which is caused because of competitive inhibitors is called competitive inhibition. The inhibition which is caused because of the non-competitive inhibitor is called a non-competitive inhibition, and the inhibition which is caused because of the feedback or allosteric inhibitor is called allosteric inhibition or feedback inhibition or end product inhibition. So there are enzyme inhibitors means the chemical substances which can have the ability to inhibit or prevent the enzyme action, and there are three different kinds of enzyme inhibitors: competitive inhibitor, non-competitive inhibitor, feedback or allosteric or end product inhibitor. Okay, then coming to each and every one of them. First one is competitive inhibitor. Competitive inhibitor. Okay, so this is the enzyme. This is the enzyme and this is the active site, and this is the actual substrate. This is the substrate, this is the enzyme, and this is the active site. Okay, so if in that medium, if this kind of a chemical substance is there, so you can see this is a chemical substance. It is not the substrate, this is the substrate, and this is another substance available with the medium, and that substance will have some structural similarity with the substrate. You can see this substance, this kind of molecule is having some structural similarity with the substrate. Okay, so when this kind of chemical substances are there in the medium which is having some kind of structural similarity with the substrate, what will do? This molecule will compete with the substrate for occupying the active site. So this is actually the substrate. This has to bind with the active site, but there are some other substances within the medium which is having some structural similarity with the substrate, so that this substance will compete with the substrate to occupy the same active site. Okay, so this is the kind of inhibitor. Since it is having structural similarity with the substrate and it is competing with the substrate for binding with the active site to the enzyme, this kind of inhibitor is called competitive inhibitor. It is a kind of inhibitor which is having structural similarity with the substrate and are competing for the same active site of the enzyme, which is called competitive inhibitor. Okay, if in that competition, if the competitive inhibitor wins, what will happen? The competitive inhibitor will bind with the active site of the enzyme, so that the substrate cannot bind with only one active site is there, and there are two competitors. One is the actual substrate, and then the second one is the competitive inhibitor. If in that competition the competitive inhibitor win, then that instead of the substrate, the competitive inhibitor will bind with the active site of the enzyme, so that the enzyme action, enzyme action will happen only if the substrate is get binded. In this case, instead of the substrate, the competitive inhibitor is get binded with the active site of the enzyme. That means the enzyme, there will not be any enzyme action. So this kind of inhibition of enzyme action is called competitive inhibition, and this kind of inhibitor is called competitive inhibitor. Okay, so competitive inhibitor is a kind of enzyme inhibitor which is having structural similarity with the substrate, and that competitive inhibitor will, since it is having structural similarity with the substrate, it will compete with the substrate for occupying the same active site of the enzyme. And if the competitive inhibitor wins in that competition, the inhibitor will bind with the active site of the enzyme instead of the substrate, so that the enzyme action is get inhibited. And the kind of inhibition is called competitive inhibition, and the kind of inhibitor is called competitive inhibitor. Okay, and the number 2 is non-competitive inhibition. Non-competitive inhibition. This is another enzyme action. So this is the enzyme. This is the enzyme, and these are the active sites. These are the active sites. This is the substrate, and this is the inhibitor. You can see in the case of competitive inhibition. So in the case of competitive inhibition, substrate and competitive inhibitor will have some similar structural configuration. But when coming to the non-competitive inhibition, this is the inhibitor and this is the substrate. There will not be any structural similarity with the substrate and inhibitor. Okay, but instead of that, even though the inhibitor is not having any structural similarity with the substrate, the inhibitor will bind with another site of the enzyme. Okay, so the inhibitor is coming and it is binding. This is the active site. This is the actual active site of the enzyme where this substrate has to bind. This is the active site of the enzyme where this substrate has to bind. Okay, so but there are some inhibitors. That inhibitors will bind with another site of the enzyme, not the active site, another site of the enzyme. So just like this, another site of the enzyme, and it is changing the active site. When this inhibitor is binding with the active site with the enzyme, it will change the structural configuration of the active site. So the active site is get changed, and since this active site is get changed in this way, this substrate cannot bind with this substrate can bind with this active site. According to the Locke and Key theory, you can see only this substrate can bind with this particular active site. Okay, but when this inhibitor is get binded with this enzyme, it is actually influencing the enzyme to change its active site so that the substrate cannot bind with the active site, and so that will inhibit. This kind of inhibitor will inhibit the total enzyme action. Okay, so this kind of inhibitor is called a non-competitive, because it is not competing with the enzyme for the active site, but it is actually changing the active site to the enzyme. Okay, and this kind of inhibitor is called a non-competitive inhibitor, and this kind of inhibition is called a non-competitive inhibition. Okay, so non-competitive inhibitor is a kind of inhibitor which is not having any structural similarity with the substrate, but it is binding with the enzyme at a site other than the active site, and once it is get binded with the enzyme, it will change the active site of the enzyme, the conformation of the active site of the enzyme, so that the substrate cannot bind with the active site of the enzyme. So the total enzyme action is get inhibited or prevented. So that kind of inhibition is called non-competitive inhibition, and that kind of inhibitor is called non-competitive inhibitor. Okay, and the final one, allosteric or feedback or end product inhibition. Allosteric inhibition or feedback inhibition or end product inhibition. So this kind of inhibition is there for a series of biochemical reaction, not for a single. In the case of competitive inhibitor, only a single step, a single chemical reaction. In the case of non-competitive, also there is a single chemical reaction. But this kind of inhibition is there in a series of biochemical, in a series of biochemical reaction where step 1, step 2, step 3, step 4, first reaction followed by, for the A and B are combining together to form C. C will be the substrate of the second step, leading to the formation of D. D will be the substrate of the third step, leading to, so that kind of series, series of biochemical reaction. In those cases, a specific kind of inhibition is there, that is called allosteric or feedback or end product. For example, so this is the first enzyme and this is the substrate. This is the substrate and this is the first enzyme. This is the active site. Okay, so enzyme substrate complex will be forming. Enzyme 1, enzyme substrate complex will be forming. Okay, this is the product. This is the product of the first reaction. Okay, then it is received by the second enzyme, and this is the active site of the enzyme. Enzyme will bind with the active site of the second enzyme, forming the second product. Okay, this product will be the substrate of the third reaction, which is catalyzed by the third enzyme, and this is the active site to which the third substrate will bind, forming the end product. This is the end product. So there are three enzymes in this series of biochemical reaction. There are 3 steps of reaction, and each of these steps are coordinated by enzyme 1, enzyme 2, and enzyme 3. This is the first substrate, the initial substrate. This is the product of the first reaction, which is acting as a substrate of the second step. This is the product of the second step, which is acting as the substrate for the third step, and this is the final product. So this is the sequence of chemical reaction. Okay, so this is normal process. Substrate is coming, first substrate is coming, binding with the first enzyme, forming the product. This is the substrate of the second step, binding with the second enzyme, forming the product, second product. This is the substrate of the third enzyme, and finally this kind of end product is formed. Okay, when this end product is formed, this end product can bind with the first enzyme at a position other than the active. This is the active site. This is actually the active site. This end product can bind with another site, not the active site. And this particular site is called allosteric site. Allosteric site. When the end product of this chemical reaction is binding with the allosteric site, what will happen? It will change. So this is the normal active site. This is the normal active site of the first enzyme. But when the end product is get binded with the allosteric site of this first enzyme, it will change the active site of this particular, this is the normal active site, it will change the particular active site in this way. This is the way. Okay. So if the active site is changed in this way, then in the second series, this substrate cannot bind with here according to the Lock-on-Key theory. Only this substrate can bind with this, the enzyme with particular this active site. But in this case, when the end product is get binded with the allosteric site, it is changing the active site in this way, so that the first enzyme, the first substrate cannot bind with the enzyme. So in the second level, in the second level, the first enzyme is get deactivated. If the first enzyme is deactivated, the product will not be forming. So the entire process will be get checked. Entire product will be get checked. So this is the kind of inhibitor. Here, the end product is the inhibitor. The end product of the sequence is the inhibitor. It is inhibited in such a way that it is get binded with the with a site, not the active site of the first enzyme, and thereby changing the the conformation of the active site of the first enzyme, so that the first substrate cannot bind with the first enzyme, leading to the suppression of the complete sequence of biochemical reaction. So this kind of inhibition is called allosteric inhibition, or feedback inhibition, or end product inhibition. So in this case, the end product is acting as the inhibitor. So the name end product inhibition, and the kind of inhibitor is called allosteric inhibitor, or feedback inhibitor, or end product inhibitor. So these are the three different kinds of enzyme inhibition. Competitive inhibition, it is for a single chemical reaction, single step of chemical reaction. Non-competitive inhibition, again it is for a single step of chemical reaction. And the final one, allosteric or feedback or end product inhibition, this is for a total a sequence of biochemical reaction. The end product of the sequence will be acting as the inhibitor, and that end product inhibition inhibitor, and so we will call those inhibitor as end product inhibition. Okay, thank you.